Okay, coming up, we've got Dr. Todd Buxton is going to talk about thermal stratification in pools on the Trinity River. Todd works on flow and sediment issues on the Trinity River for the TRRP and is currently investigating flow effects on temperature stratification in river pools, development of an acoustic technique for bed load monitoring, and evaluation of Rush and Indian Creek deltas and their capacity for rearing juvenile Chinook salmon. Todd completed a 4-year enlistment in the U.S. Coast Guard before starting his career in river and salmon restoration in 1994. His work has mainly focused on the interties between sediment transport dynamics, stream flow, and biological populations in rivers in the western U.S., Alaska, New York, and Costa Rica. Todd has earned a BS in watershed analysis and restoration and an MS in watershed management from Humboldt State University and a Ph.D. in water resources from the University of Idaho. His academic research included developing and testing an equation that predicts entrainment of waterlogged wood in rivers, streambed packing effects on sediment mobility, relative stability of salmon reds in ambient streambed areas, and salmon spawning effects on hypereic flow and marine nutrients from salmon in streams. Todd's free time is preferably spent building wood structures of any kind and caring for the land where he lives along Browns Creek. And with that, I'll hand it over to Todd.
Okay, thanks for being here. This work was performed in the field in 2020, and the modeling was completed in 2021, and this paper was published in Hydrological Processes Journal in 2022. This is the start of a multi-year study that continues, and you'll see where it's headed from this initial step. I want to recognize my co-authors listed there, and then a whole bunch of different folks for helping out in the field. So start with the basics. Thermal stratification is simply the arrangement of water temperatures in a thermocline with warm water at the surface and cold water deeper. And stratification is important, as you've been hearing, because it gives species access to a wider range of temperatures than if the water is mixed and temperatures are homogeneous. So most folks are familiar with thermal stratification in lakes. Stratification occurs in rivers too, as you'll see. So our goal was to identify the mechanics of stratification in rivers and validate a model for simulating them. And our approach was to document flow and temperature conditions that prevent or form thermal stratification in deep river pools. That 2 meters will come up several times. It's an important depth. We validated a 3D model for predicting critical flows for stratification and temperature gradients in pools under variable meteorologic conditions. And then we used those field and model results to explain the stratification process and its impact on species. So our study was conducted in 2 pools. One pool is located in the Upper Trinity River, UT pool. And in that area of the river, there's natural hydrology, although the channel is quite impacted by past mining activities. But the natural flows that occur there are key to stratification that we measured in this UT pool, as you'll see. And then you've got the water storage reservoirs, Trinity and Lewiston dams, and then at the downstream end of the Trinity River Restoration Reach, which is 40 miles long, is Pear Tree location, and that pool we studied is PT pool. So here's a curve showing the historic daily average flow at these locations. The blue line is the UT pool, and then the other colored lines are the flow at Lewiston. The orange line is the pre-dam discharge, which was in summer about 150 CFS on the Trinity River. That was twice as wide as it is now. The channel is narrowed because of flow regulation, reducing flood magnitudes. And so now what we see on the restoration reach at Lewiston is a summer base flow of 450 CFS, but we released those Hoopa boat dance flows in August, and we released Klamath augmentation flows. And so if you do the daily average through the summer period, we have a discharge coming out of Lewiston that's upwards of 700 to 900 CFS in comparison, again, to that 150 pre-dam. So that higher discharge in a narrower channel. So that elevated base flow that's released at Lewiston is intended to provide fish cold water habitat that they're blocked from accessing because of the dams in the upper watershed. But you'll see that the temperatures in the upper watershed are actually quite warmer than we provide downstream of the dams now. So there's significant opportunity for improving thermal management in the river and benefiting fish, frogs, and other creatures that you'll see here. So here are the study pools, UT pools, the top two panels, and PT pools, the bottom two panels. And both pools were instrumented with pressure transducers and temperature loggers at the inlet, outlet, and in the pool body. And then we also constructed stringers of temperature monitors by weighting a cable with a float on the top and mounting temperature monitors between those 2 points. So we could monitor temperatures every 15 minutes throughout all depths in the pool with these stringers, and you'll see the results from those. But we also measured vertical velocity and temperature profiles. The temperatures were measured with a tidbit attached to the acoustic Doppler velocimeter that was used to measure the velocities in 3D. So UT pool is about half the size as PT pool, just keep that in mind. The max depth of base flow, summer base flow in both these pools is about the same at around 5 meters. And then the summer base flow at PT pool is about 14 cubic meters per second. And the summer base flow at UT pool is about a half a cubic meter per second. So we release water so that there's 28 times more discharge in the restoration reach than there is upstream where the salmon would access to hold through summer. And the reason we release that extra water is to provide the cold water, but I'll show later that that's really not necessary. So here are the results for UT pool. The top figure shows the temperatures from the stringers, and there were 2 stringers installed. They both show the same results. So here's the bottom color is the temperature measured at the pool bottom. And then the top colored lines are the temperatures measured at the pool inlet, outlet, and near the surface. And so you can see, the gray line is the air temperature measured every 15 minutes, and the daily average air temperature is that orange line. You can see that stratification, which is defined as a temperature difference in a vertical being 1 degree Celsius or more, formed first on June 28th, and that formation lasted until November 11th, although it was destroyed and reformed on a daily basis, as we'll see. The next figure down shows the duration of the day with stratification formed, and that was up to 13 hours a day with an average of 8 hours. So there was temperature diversity produced in UT pool for most of every day. And the degree of stratification, so the difference between the min and max, was up to 8 degrees Celsius. So that gives juvenile salmon quite a range of thermal variability to use to their advantage in putting on weight. So remember, a juvenile fish will feed to satiation and then seek warmer waters for digestion to put on weight. And then we observe this in the pool where towards the end of the day, when their digestion is completing, they go to the bottom depths of the pool where it's colder to slow their metabolism. So the body doesn't start eating the weight that they just put on. And so after that cold water period for these juvenile fish, they go back out and feed and then spend their night back in the pool. That's what we observed. It probably works differently in different places, but that's what we saw in this pool. So that 8 degree temperature difference gives those fish all the habitats they need for that cycle. So stratification in the study formed in the pool between 10 o'clock in the morning and 2 o'clock in the afternoon. and it was destroyed between 1 o'clock in the afternoon and as late as almost midnight. And that variability results because of differences in meteorology. So warm water forms stratification earlier, and cold weather can make it form later in the day. So incipient stratification formed on June 28th. It had discharged about 1 cubic meters per second. And for you CFS thinkers, there's 35 CFS per cubic meter. So you can do the math. So if you notice, the stratification formed on the day when the temperature variability delivered to the pool was quite a bit lower than in previous days. So the reason for that is, while you do need temperature variability for stratification to form, you also need a sufficiently low discharge. Because if your discharge is too high, the mixing will be too high and those temperatures will mix. So it's the density differences in the different temperature water delivered to a pool throughout the day that is responsible for the possibility of stratification, but you have to have critical or subcritical discharges in order for that stratification to form. So once stratification formed, the pool transitioned into a period of daily formation and destruction of that stratification. It happened every day from June 28th until the end of the stratification period, and it was quite regular, with the exception of when wildfire smoke came into the area and lowered all temperatures by about 6 degrees Celsius. And also when there was some weather systems that came through and just delayed the formation, but it still formed every day in subcritical flows. So looking at that process in detail, here you see the inlet water is shown by the dotted line, and it in nighttime is colder than all other temperatures in the pool and at the outlet. And so as time marches through, the sun rises, and once the solar radiation first hits the river bottom, you start to see that inlet water warm. That water warms to a degree that is above all other depths in the pool after the sun is in the entire river bottom. So you have direct solar radiation warming the entire river bottom, and you have warm inlet water buoying to the top by convection. So that warmer inlet water, though, is mixing with the bottom depths in the pool by diffusion. And so what you have is that cold water body that's stored overnight and formed, slowly leaking cold water downstream. So you're also seeing different depths in the pool increase in temperature as a result of that diffusion process. And so you start to see a temperature gradient that strengthens throughout the day until the heat of the day, which is right before shade starts to hit the river bottom from trees and mountains. And so once that shading begins, that inlet water cools and sinks and starts to displace the temperature layering in the pool. That stratification then is quite rapidly destroyed because of that mixing that occurs from the colder inlet water sinking through the depths of the pool. And that continues until just before sunset. And at that point, the stratification is destroyed. The pool inlet water is again cooler than all other depths in the pool, and you restart the cycle. Now, that's kind of a cool, nerdy thing, but what's important to downstream habitats here is that during the time of day when the inlet water is colder than the pool is releasing warm water downstream. And then during the heat of the day, that warm water that's transiting the pool in its upper surfaces, as you'll see in the next slide, is displacing that cold water that was stored overnight. So it's releasing cold water downstream up to a degree Celsius. And then the cold water or the warm water starts to be released again towards the end of the day. And so you can imagine that if all your pools that are deep in a river stratify, you have your temperature management right there. It's got a temperature regulating effect that could be quite significant. So here are the temperature and velocity profiles. You can see that the profiles were mixed during that slow mixing period when the diffusion process is occurring. There is a faster top water flowing over the pool in the thermocline area of the temperature profile. That water is at a blistering 12 centimeters a second, but below it, the water is almost still around 2 centimeters per second. So again, faster top flow, slower bottom flow. Where are the adult salmon? They're in the slower bottom flow. So this cold, still pool water body is ideal for spring Chinook holding. And that's what forms here with stratification. And you can see the temperature profiles, very strong diversity in temperatures, not only with depth, but time. And the reason those profiles shift towards warmer water is that they were measured at different times of the day. So the times of day that these particular profiles were measured are indicated by the red bars. And you can see that as the day progresses, the pool warms, the thermocline shifts to the right towards warmer water. Also note that, if this is the model for what we're trying to create in the restoration reach in terms of temperatures and pools for spring Chinook holding, we have a temperature range here of 18 to 24 degrees. But look at the bottom temperatures. They're consistently 18. We got the temperature diversity for the juveniles. We have the cold, stable water for the adults. Pretty ideal. So towards the end of the stratification period, we had air temperatures that were actually below freezing, but the daytime temperatures increased to around 10 degrees Celsius. So again, it's the difference in density of water delivered to the pool in the diurnal cycle that causes or enables the possibility of stratification. And then with subcritical discharges, the stratification forms as it did for the last time on November 11th. But thereafter, the temperature range decreased. It was more uniformly cold. And if you look at the density curve by temperature for water, there's very little density difference in those temperatures. And so you need an even lower discharge than the critical discharge that started the stratification process in this pool in order for stratification to form in these conditions. And that's why after November 11th, the temperature range decreased, the discharge stayed low, but the stratification didn't reform. And then the flow increased above critical, and that was the end of the period. Much simpler story at PT pool. Here's the result for that. You can see in the left figure that there's a single line for all those different temperatures with depth. It's because they're all the same temperature. There's no diversity. We do have diurnal fluctuations in temperature at this pool, and it's at the downstream end of the restoration range, where if there's going to be any possibility of stratification, it's going to be there because you're going to have the greatest diurnal variation in water densities. But still, we see a single line because all the temperatures in this pool are the same. Here's the velocity and temperature profiles at PT pool. The reason for the uniform temperatures is high flows. And so what we have are velocities up to almost a meter per second. That's too high even for the spring Chinook holding. So we think we're doing well by the adults by releasing all this water, but I would argue we're doing the opposite. And so again, here's the ISO heights measured in this pool because it's fully mixed. There's no temperature diversity in the vertical. There is through time because of that diurnal variability. We can do a lot better than this. And I think there's pretty strong justification for managing for stratification when you compare the results between these two pools. Remember, we've got 28 times higher flow at PT pool, but the daily vertically average temperatures that we measured through the heat of summer at both locations were pretty much the same. So if our goal in releasing these high temperatures, regulatorily speaking, is to meet temperature targets, we could do so with a fraction of the water that we're spending on it now. And that water could be used for other environmental benefits at other times of year. So moving on to the modeling, we used a model developed by Yang Lai. He calls it U2 RANS. It stands for unstructured and steady riddles, average Navier-Stokes equations that were used to solve the mass, momentum, and energy conservation equations in 3 dimensions. And here's the mesh shown below. The left figure is a plan view of UT pool, and then the underneath view of that same pool showing all the computational cells is shown on the right. So those equations were solved in an unstructured 3D mesh. It's bounded by the water surface at the top and the pool bathymetry at the bottom. And in this verification study, the PT pool mesh had 230,000 cells, and the UT pool mesh had 152,000. We did a mesh sensitivity analysis, and these were appropriate densities of mesh to get mesh independence in our results. So here are the results. The dots are the measured values, and the lines are the modeled results for UT pool. And the model predicted the observed temperatures to within 0.5 degrees Celsius, 85% of the time. And then at the initiation of stratification, we saw an error, model error of almost 2 degrees Celsius. And we think the reason for that is that hyperrheic contributions of water and different temperatures that were in the surface water was affecting the results, but that's a guess because we didn't measure hyperrheic flow or temperatures at the site. But nonetheless, the model results trace the measured values quite well. And then at the PT pool, the observed isotherms were predicted to within 0.25 degrees Celsius. So here are the results for seeing how well the model could predict the critical discharge for stratification, and did quite well here too. The model estimated the critical discharge is about 1.03 cubic meters per second versus the 1.08 that we observed. And then we used the model to predict what the critical discharge at PT pool would be under observed temperatures, and that was around 2 cubic meters per second or 70 CFS. So the 2 times higher critical discharge estimated for PT pool reflects its 2.5 times larger size for dispersing inlet velocities. So if you have a larger pool, it can take a higher discharge for the critical value because there's more capability for dispersing that inlet water and preventing mixing at a higher flow. So other factors that might influence that critical discharge are pool roughness or pool shape, the location of the inlet jet relative to the deepest depths in the pool. And we're studying those now in an expansion of the study that I'll go over in a little bit. So I played around a little bit with what that 70 CFS critical flow at PT pool could mean. And so how much smaller is the Trinity River with dams versus before dams? I used the 1960 pre-dam aerial photograph and the 2020 post-dam, I say hydrograph, I meant aerial photo. aerial photo. So I measured the active channel width in both cases, every 1/10 of a mile, all the way from the dams to the North Fork Trinity River, 40-mile section, and found that the average reduction in active channel width was about 45%. And so the scaling's uncertain. I'm just playing here. But if you reduce the pre-dam flow observed in summer by 45%, you come up with an average summer base flow of around 70 CFS. So going back to natural flow regimes and what might be needed on the Trinity River in summer, we have a template. We have pre-dam flow records. We know that the species prefer warm temperatures and they need the diversity of temperatures for their life cycle. So if we were to reduce the summer base flow appropriately to reduce the effect of freezing out species and providing them with only one type of habitat for a life cycle that is completely diverse, I think we'd be more successful. So in summary, diurnal temperature variations occur in the Trinity River, but unnaturally high regulated summer flows cause mixing that generates spatially uniform temperatures and high velocities in pools. And these conditions prevent juvenile salmonids from accessing warmer and variable temperatures to maximize growth, and adult salmon from holding in cold, deep water with slow velocities. If stratified pools were available in the restoration reach, they would provide adult salmon cold, deep, slow water for holding in daytime until the entire river cools for migration to resume at night, as those temperature variabilities in the diel cycle show. And actually, that's what Moffat and Smith observed pre-dam on the Trinity River. As it is currently, we release these high flows, we freeze out the river, and then that, as Eli showed, those temperatures that are provided by the high flows from the dam don't really do much downriver. So we still have thermal barriers to migration forming in the Trinity River, despite the expenditure of all this extra water. Instead, what we could do is release summer flows that promote stratification, enable the river to actually cool more at night because there's less water to cool at night. And so then the fish could use the deep stratified pools in daytime rest, wait till evening when the river cools and resume their migration. Then we wouldn't be freezing out the river unnaturally, and the fish would be on their way. So thermal stratification was up to 8 degrees Celsius at 0.5 cubic meters per second at UT pool in the heat of summer, when the daily vertically average temperatures were the same as in PT pool at 14 cubic meters per second, when stratification did not occur. And so that to me says lowering dam releases to stratify pools can provide warm and cold water habitat with much less water than is currently released in summer. And the bottom line is the summer flow regime on the regulated Trinity River is completely outdated. We've got science that supports lowering those discharges, warming the river, providing the temperature diversity, not just for fish, but for frogs and turtles and things too. And then there's benefits to the food base, right? And so we would have more food for our juvenile fish to eat, and then the right temperatures for them to grow larger. And so as it is now, we release the high flows, we freeze out the little fish. They're smaller going out under the rod than they were even before the rod, and survival to adult tends to increase with juvenile fish size. So our aim to benefit future adult salmon populations with our water regulation and releases is actually probably doing the opposite. So requirements for thermal stratification that we discovered are that low discharge includes low discharge to prevent thermal mixing, large pool volume to disperse fluid momentum, deep water to attenuate solar radiation, and inlet temperatures that diverge in space or time. In our study, the temperatures diverged in time. Conditions where stratification might form with temperatures that diverge in space would be, say, a pool near a cold water seep or a hot spring or something like that. I neglected to say why the 2-meter depth was important earlier. And the reason it's important is because at a depth of 2 meters, the exponential decay in solar radiation is such that the warm water is kept mostly towards the top surfaces of a pool. And so that's coincidentally, or maybe not, where spring Chinook tend to hold, which is in pools deeper than 2 meters. So deeper pools stratify more, and the depth provides cover, and that's what the adult spring Chinook like. So as I mentioned, this is an ongoing study. This is a big wordy thing. I'm not going to read it. You can read it if you want. But basically, we're going to expand the study to all pools that meet the depth criteria for spring Chinook holding in the restoration reach. Do the 3D modeling, which requires a number of different models to inform the 3D model for it to produce results. And it includes 2D modeling for the water surface, 1D modeling for subdaily temperatures that are unobserved flows. And all of that modeling is complete except for the subdaily temperatures. I just completed that model yesterday and it's being calibrated now. So we're off and running. In the end, what we're going to have are habitat holding curves for each pool that meet the depth criteria for the springers. We sum those areas at a given discharge, and we can say, okay, at this flow, you've got holding habitat for this many adults. Our goal is to provide enough holding habitat for the escapement goal of spring Chinook. And so that's where we're headed. And here's references to this study and to Young's model. I'll take any questions. Thanks.
Hey, Todd.
Hey.
This is probably the third or fourth time I've seen you present your results, and every time it's very thought-provoking, I think, for me. Is there, with the air temperature between the upper Trinity and that pear tree, did you see a larger daily max min variation at the higher elevation versus down a lower elevation at pear tree?
Yeah, the air temperatures ranged more widely at UT.
Would that require then an even lower critical discharge potentially when you're in a lower elevation in order to get the pool to stratify? I mean, because you're getting the assistance of a very, high daily variation in air temperature.
Yeah, I think there's two things going on there where the temperature monitors for air temperature were located in trees next to the river. And so you have the natural heating, cooling that occurs on the UT pool. And so you didn't have that perpetually cold water coming down the river and affecting the air temperatures that you had at PT pool.
Right.
Yeah. Yep. So that could be part of it. The other thing is that the critical discharges on the main stem are going to be larger because the pools are larger. And so just for reference, the PT pool is 5,000 square feet or something like that in area with depths 2 meters or above. There's pools in the main stem that have those areas of around 35,000. So the low critical or the 70 CFS critical discharge at PT pool isn't representative necessarily of what the critical discharge will be at other pools. So that's where a variable flow regime in summer that's low enough to enable stratification could be another impetus to push fish upstream.
Thanks.
Yeah.
Are we supposed to say your name for the recording thing or anything? Okay. I'll say it anyway. Mike Orcutt, Hoopa Valley tribal member. My first comments are that, as a Hoopa Valley tribal member, this is the first time I ever heard any, concern about the boat dance flows, which are not always in August, but sometimes in September. I've never heard anything about it being detrimental to anything. First time I've ever heard of it. I would note that in the litigation that the tribe and Interior were defendants in, that the parties that brought Westland Water District and San Luis and Delta Minnesota Water Authority, there's a stipulation in there that they don't even contest the boat dance flow. So to hear this about that is a little bit surprising. And if it is, I request that the right folks in the Bureau of Reclamation contact us for that and see if there is a concern about that. The second thing I have is the spring Chinook. I'd start with that. Yesterday, I'm not sure it was a presentation, but it definitely was at the Lewiston Hotel. It was pointed out that the spring run, and I'm not going to get into a debate about the numbers, but the best available science for the spring run are pretty regularly met for as far as the goals are met. The Hoopa tribe has been a major party and a proponent of getting some management and conservation standards in place for spring run Chinook. It's presently being petitioned for listing. There's some things I have problems with the regulatory agency with that, but nonetheless, that's the case. So I guess I'd just wonder what's the purpose of it if we're making most of the escapement goals. And then the last thing I would say is I'm not sure the science part of the program is supposed to be in the business of saving water for the Bureau of Reclamation. I think we have, as a tribal resource manager and scientists, have a hard time and a real struggle with this whole saving water. It was said yesterday, okay, the geomorphic flows can be met in one day. The temperature, this cold water is an impediment to fish production, which I don't think we know that yet. I think there's a lot of analysis, and perhaps the limiting factors analysis will be the proper forum for that. So what I would end with would be that the spring run is something I fished for a good 35, 40 years now. And I've observed a lot of different things with that. And I think it's more some of the things that we have also promoted, getting a little off track, but is the integration of the hatchery and a natural production, which is an artifact of a time frame when the program was trying to save money. And so that's why there is no integration of that management with the mitigation responsibilities at the hatchery with the natural production. And so a lot of things that need to be fixed here, but I'm not sure I see the picture as far as the spring run is at. Thank you.
Thanks, Mike. Yeah, let's be in touch to talk more about Hoopa boat dance effects on some of these ecosystem processes and temperatures that have been brought up. And then in terms of the effect of non-stratified flows on the springers, I would imagine that if you're an adult fish that has to hold in cold water that's fast, you're going to have lower fitness when it comes time to get on the spawning grounds. I got to run because I see Kyle about ready to kick me out, but that's my first thought on the effect of non-stratified flows during height discharges on the main stem Trinity.